Well, thanks very much, Terry, for the introduction, and thanks, Luan, for the organisation of the lecture, and thank you to all of you for coming to this lecture. So what I want to do is to talk about systems biology and give you my view of what systems biology should be, because I think a lot of people disagree about this scaffolding. People haven't agreed what kind of scaffolding we need. So what I want to do is to give you my views as to what it should be. And I'm going to do that by using some examples of work from my group in Auckland at the Bioengineering Institute. And these are projects which are ongoing. So the work I'm going to be talking about, I'm going to pick examples which I hope will illustrate points that I want to make from work that's ongoing in my group. But before I do that, I want to just show you this. So the World Economic Forum recently as last month, published their list of the top 10 emerging technologies for 2012. So they do this every year. They publish a list of what they think are going to be technologies that have the potential to make significant economic benefits and which have the potential to increase quality of life. And in their list is systems biology and computational modeling and simulation of chemical and biological systems. Amongst things such as low carbon technologies, synthetic biology, so the new name, if you like, for genetic engineering, systems biology. So what is systems biology? Well, I am an Englishman and a scientist, and I think like all good Englishmen who are scientists, I think that the ultimate repository of scientific wisdom lies in the journal Nature. So Nature has recently, what is systems biology? Nature has recently set up a new journal called Molecular Systems Biology. And this is their definition of what systems biology is. They say that molecular systems biology is an integrative discipline that seeks to explain the properties and behavior of complex biological systems in terms of their molecular components and their interactions. And I think that's a wonderful description. But I think that's a wonderful description that many cell biologists would say is a description of what they've been doing for decades. And I think it's a wonderful description of what a lot of physiologists would say their discipline has been doing for decades. And I don't think there's anything here that really singles out something new, this new scaffold, something new about systems biology, which is going to allow progress that hasn't been possible before. So I think Nature has let us down slightly. So what would one do then if Nature has let one down? One would go to the internet, of course. So if you type systems biology into your favorite web browser, this is what might come up. So systemsbiology.org seems very promising. Systemsbiology.org says systems biology is an emergent field that aims at systems-level understanding of biological systems, which is, again, hard to argue with. This is actually from Kitano's Institute for Systems Biology in Japan, and so I think they might be forgiven for this rather sort of tautologous statement. There may be something is lost in translation, perhaps. So, again, then one wants precision, and for precision, one turns to the Swiss. So the Swiss have recently put in something like... I've put that up too quickly, haven't I? Let me tell you, so systems biology is an emerging field in biology that's still attempting to define itself. Well, I mean, you couldn't get closer to the truth than that. So the Swiss government has recently put, I did the calculation, something like 160 million Australian dollars into a systems biology program that they call Systems X. So a program across many institutes in Switzerland. So a lot of money, a lot of resources is going to this area that they haven't yet really defined, which is, confidence, I suppose. There are some, in all seriousness, there are some definitions that I think are useful and which I'll start to use to work towards what I think is important in systems biology. So Leroy Hood in his institute in Seattle. So Leroy Hood, one of the pioneers in RNA, in automating RNA sequencing. So he has an institute for systems biology in Seattle, and he talks about systems biology as being the study of an organism viewed as an integrated and interacting network of genes, proteins, and biochemical reactions which give rise to life. And I think there are two important things here. One is that he talks about a network, so we have this idea of putting things together in a network structure. And secondly, he talks about the biochemical reactions, and so that gives us an idea that we may be interested not just in some structure, but in the dynamic interactions on that structure. And another example that I like is from about 5 years ago now, is from an issue of Proteomics, a special issue that was put out which we contributed to. And in the editorial, they described systems biology as an attempt to structure biological knowledge into hierarchical levels from gene products to whole organisms, and to look at the rules which will determine, which will allow us to navigate between these levels. And I think that other idea about this sort of hierarchy is very important. So for me, my primary interest is in the sort of grand challenge, which I think is a grand challenge for all of systems biology, which is to try to relate genotype to phenotype. So to try to relate the molecular information we may have about a biological system into knowledge, understanding about the phenotype, about the behavior of the system as a whole. And this is a challenge. There's no question about it. This is a challenge. How does the heritable information encode the phenotype? What can this tell us about disease? And this is a particular challenge because we know if you take a purely genomic approach, where one looks for statistical associations from genomic data to try to understand the variation in disease, for example, so-called genome-wide association studies, although this is an approach that has a huge promise and has still got huge promise, in complex diseases, cardiovascular disease, diseases of the immune system, you can only explain a very, very small fraction of observed variation across a population, phenotypic variation across a population, in terms of identifying loci in the genome which account for that variation. So there's something that's missing, one might argue. What's even more apparent is that we have absolutely no ability to predict the outcome of variability at the genomic level in terms of its impact, its effect at the phenotype, at the level of phenotype, the level of function of organisms. We simply can't do that at all for the most part. And so my view is that we need models to connect genotype to phenotype. And so although my training is in physics and maths, I now live in an engineering institute. And so the engineer's approach is to take a system, to perturb it in a number of different ways. And from observing what happens when you perturb it in a number of different ways, then you can build a model which relates those perturbations to the resulting changes. And so I think that systems biology takes this approach. Systems biology says we take an organism, or a biological system, we perturb it in a number of different ways to explore the range of behaviors of that system, and we use a model to connect the perturbations we've made to the phenotype. So let me give you very briefly the overview of a project that I'm involved with, which is taking this very approach. And this is called the Virtual Physiological Rat Project. So this is a recently funded NIH Systems Biology Center. The NIH has moved into this area with a lot of resources. It's based at the Medical College of Wisconsin, which also houses the rat genome database. And the ambition for this project, which has only been running 6 months, so I can't give you any results yet, but the ambition of the project is to use models, computational models, to link genotype to phenotype, and in particular to focus on cardiovascular disease and to use the rat as an animal model for cardiovascular disease. So what we are doing in this project is we're taking different rat strains which have known genotypic variation and which have measurable known changes in phenotype associated with different aspects of cardiovascular disease, and we are building computational models that describe the cellular processes involved, put those cellular processes into models of tissue, be it cardiac tissue or renal, and by using a number of different rat strains, we're able to build models which relate the changes at the molecular level, the genotype changes, through to explaining or being consistent with the phenotypes. But the key thing about this project is that we don't stop there. What we then use is the models to predict the outcome of as yet unrealized mutations. So we use the model to predict what happens if we make a new genetic mutation, and then using the resources at Medical College of Wisconsin from the rat genome people, we then engineer a new rat model, a new animal using knock-in or knock-out of particular genes, and we see whether our prediction is actually matched by reality. So if we can do this, then I think this opens up what's perhaps the most important aspect of this, and is the sort of second meaning of my title about a personal view. Because I think if we can do this, then we can really open up an avenue towards personalized medicine. So if we can use computational models which genuinely provide a mapping between genomic and phenotypic variation, which relate changes in genotype to measurable differences in phenotype, then we can understand the causes, the mechanisms underlying disease. This allows us potentially to approach another great question, which we have limited ways of understanding, which is how do we understand why a drug will work so efficiently for some patients and so poorly for others, or maybe toxic to some and not to others. So the way we do this, of course, is to use the models to link, in the case of pharmaceutical compounds, drugs, to link the mechanism of action to predict the likely outcome, but not on the basis of some averaged model, but on the basis, so we're trying to link the drug to the response, the phenotypic response, but not in some sort of averaged way. We use the model to do this, but then we can reparameterize models for different genotypes. So if we can do all this, then I think we have an approach to personalized medicine which is predictive, which predicts the outcome of particular drug therapies for given phenotypes. So that's my overview, my motivation for this area. So now to the personalized view. My personalized view of what it takes to be able to do this, what I think it's likely to take to be able to do this, is that we do need this concept of networks that I referred to before. We need the idea that we're including many pathways, many interacting components. And so there is a key need for data integration for this kind of enterprise. But secondly, I think something which is commonly overlooked and which I will talk about is the need for that data to be specific to the context, to the tissue, to the cell type. And I'll show you an example of why I think that's so important. And then the third thing is that for these models to be predictive, and that is the key word for me, for these models to be predictive, I think that it is extremely important for them to be mechanistic, which is to say, to describe the biochemical mechanisms going on in cells, the biochemical interactions between molecules in cells, to allow us to have confidence that we can extrapolate beyond the data that we've used to build the models. So I'm driven by this idea that we need to develop increasingly systems-wide models of cellular pathways, regulatory processes, which are mechanistic and which are built from data specific to a given tissue, a given disease state. So I want to give you three examples. The first one is going to be in some of our cardiac work, looking at signaling mechanisms, and this is going to focus very much on, this is not going to be very network focused, this is going to focus on what I mean by mechanism, just to give you a view of what I mean by mechanism. And it's also going to touch on some other aspects that I think are important. The second example, which I'll only touch on quite briefly, is what's now becoming rather a conventional approach. So this is dealing with developing prognostic markers and tools, in this case for cancer, but I want to touch on that topic for a reason that will become apparent, I hope. And the third one is to look at some work we've been doing in signaling pathways in skin, and this is really focused on the necessity of looking at a particular tissue type, so not looking just at some isolated cell line, but actually looking in the tissue itself. And I hope that these examples are also going to illustrate the application of systems biology to some basic science questions, basic research. So the cardiac work is really motivated by simple questions about, how does this work? The second area is going to talk more about translational research, about actually trying to develop some tools, some diagnostic tools which may be useful to a clinician. And the third one is really about discovery science. So it's really about learning new things about the biology. So the first of these examples is in cardiac hypertrophy. So I will give you a quick overview of what cardiac hypertrophy is. So it is the increase in mass of the heart without any cell division. So in athletes who are training a lot, in pregnancy, this is a perfectly normal response to an increased demand on the heart, an increased workload on the heart. The heart grows, the muscle mass increases, not through cell division, but by each cell increasing in size. But it's also a maladaptive response. So in pathologies such as heart failure, where the heart is not matching the required cardiac output, this is a response in which pathways kick in and the heart grows in this way, hypertrophies, in a way which cannot compensate for the increased demand, or rather the failure of the heart to meet demand. And there are associated risks of sudden cardiac death, so it's of real clinical significance. From a sort of cell biology point of view, you can mimic, you can drive hypertrophic growth of cardiac myocytes simply by addition of a hormone. So a hormone such as endothelin-1, angiotensin-2 will, as shown in this figure, you've got some cardiac myocytes under the confocal microscope, and if you add the hormone to your dish, you'll see that the cells will grow, will just get larger. So there are other signals which are involved, but I'm going to specifically look at this pathway. So things like mechanical stretch are also involved, but I'm going to specifically look at these hormonal pathways. Now, I also need to tell you a bit about calcium in the heart. So calcium is crucial in heart cells, as in many different cells. And one of the reasons that calcium is crucial in the heart is because it is the messenger which couples electrical activation of heart muscle to its mechanical contraction. So if you look at this diagram, if you look at the bottom part of it, in response to electrical activation of, so this is the cell membrane I'm schematically illustrating here. This is a calcium store, the sarcoplasmic reticulum. In response to a wave of electrical activation moving through the heart muscle, this activates calcium channels, and a little bit of calcium enters the cell. That little bit of calcium triggers a much larger release of calcium from these internal stores. And that much larger amount of calcium, that transient, activates the contractile apparatus in the myofilaments and causes the cells to contract. That calcium is also sucked back into the calcium stores through a calcium pump to return you back to your resting levels. So calcium is coupling the electrical activation to the mechanical contraction of these cells. Now that's a process that's well understood and well studied and also well modeled. So lots of mathematical models describing in some detail components of this system. But calcium in the heart, as in many other cells, is doing all kinds of other things. And one of the things that it's doing is shown in this top figure here. So calcium is also a second messenger involved in mediating that hypertrophic signal. So if you have a hormone, such as endothelin, which arrives at a receptor, a G protein coupled receptor, that leads to production of IP3. The IP3 binds to IP3 receptors in other calcium stores, which releases calcium. And that calcium is what is known to signal to other proteins, so calmodulin, which senses calcium, and calcineurin, which activates a transcription factor called NFAT to be translocated into the nucleus, where it switches on gene expression. So I'm simplifying a rather more complicated pathway, but NFAT is one of the key triggers in the nucleus. Activated NFAT in the nucleus is one of the key triggers for that hypertrophic response, so for the cells to start growing. So the question is, well, how can calcium be doing these two things at once? How can this signal stay distinct from that signal? How can this signal not just always trigger hypertrophy? How does the cell keep these two signals separate? And this kind of question comes up again and again in, for example, in looking at calcium signaling, because calcium is so universal as a second messenger. Mike Berridge, who was one of the first, well, he was the first person to really understand the role of IP3 in calcium signaling. And he said that the question I'm most frequently asked about calcium is how a simple messenger can regulate so many diverse cellular processes with much hand-waving. This is my emphasis with the underlying. But with much hand-waving, I usually speculate that such diversity might be achieved by operating through different amplitude, spatial, or temporal modes. And surprisingly little is known about how this might work, how different amplitude, spatial, or temporal modes may actually work. And so we've set out to ask that basic science question: how is calcium doing these two things at once? So first of all, we need to ask, well, how is this hypertrophic pathway controlled? IP3 to release calcium, calcium to calcineurin, calcineurin to activate the transcription factor NFAT's translocation into the nucleus. How is that controlled? It is known that if you use different hormones, the responses are quite different. So why is the response to endothelin-1 quite different to the response to angiotensin-2? And in particular, the question we're interested in is, well, how can this signaling process be distinct from the beat-to-beat calcium transient that you get from excitation-contraction coupling? So this, every time the heart beats, you've got this large calcium transient, which is coupling the electrical activation to the mechanical contraction. So how is it possible these signals are distinct? And so we build mechanistic models of each of the steps in this pathway. So a mechanistic model in this case for the G protein coupled receptor. So I have different states of this receptor with ligand, so that's going to be endothelin-1 in a minute, but ligand not bound, ligand bound to the receptor, and the various other states of this protein that we know about. And we model transitions between these different states using the law of mass action, for example, which is just a sort of a simple mathematical formulation that describes how fast you move between different states, for example, if you have two molecules interacting with each other. And from that, we develop ordinary differential equation models which describe the rates at which this system works. So the rates at which we are moving between these different possible states of the receptor, the rates at which, for example, IP3 is being produced from PIP2. And so mechanistically, we describe these biochemical interactions and reactions to have a dynamical ordinary differential equation model of the cell. And it works. You can build these models and you can parameterize them and show that you can fit your model to some data, not enormously surprisingly, and you can do this in terms of time course. I'm showing the IP3 concentration as a function of time. And you can do things like dose response curves, and if you squint, you might agree with me that this sort of fits, but you know. But the point is that we haven't used these data. This is not a curve fitting exercise. We haven't used these data to build the model. We've built the model and then we've validated it by seeing whether it's consistent with the data. So we can build these kinds of mechanistic models, in this case describing the G protein couple receptor and subsequent production of IP3. That's fine, but one thing that I mentioned is that endothelin-1 and angiotensin-2 have quite different responses. And so we can analyze these models in very nice ways. So we can, for example, do sensitivity kinds of analyses. So if we look at this kind of transient showing the IP3 concentration changing over time, we can analyze this model, which is now a set of differential equations, to determine which parameters in the model are responsible for determining the peak, what parameters in the model are responsible for determining the time to peak, the decay time, all these sorts of things. And if we do that, we identify some key points in the model which are responsible for determining those characteristics of the behavior of the model. And we can very nicely show that we can change those values and we can fit now the transient for angiotensin-2. Now this may not seem at all surprising, but actually these transients are on completely different timescales. So if you plot them on top of each other, angiotensin-2 is a very rapid response, whereas endothelin-1 gives you a very long response. And what's very nice is that none of the pathway has been changed other than parameters which are to do with the ligand binding to the receptor itself. So it makes perfect biological sense that you're not changing anything else in this pathway. All this stuff is consistent and conserved, and the same cellular mechanisms are used, but we're changing parameters which describe how the ligand interacts with the receptor. So I show you this because I wanted to get across the idea of what I mean by mechanistic modeling. And I think this kind of modeling is needed to be able to predict, in the way that I've described, predict the outcome of interventions as I've described. And so we can do that for different steps in this pathway. And what's very nice is we can do this in a modular way. So we can build models which describe different components. So the G protein couple receptor, for example, we can build models that describe the calcium flux through the IP3 receptor. And using some technology called CellML, we can build more complicated models of this pathway and potentially of multiple pathways by having mathematical representations of the mathematical model of these individual biological pieces. So I'm not going to go into the details of this, but CellML allows you to have those components and to assemble them into increasingly complex models without having to look too much under the hood as to what you're doing there. So a lot of effort into developing technologies around. So CellML is essentially it's a markup language to describe these mathematical models, and it allows this modular form of how we go about building our models. When we get to the final part of this pathway, which is the regulation of transcription, so remember that the sort of key readout here is that when NFAT is translocated into the nucleus, this transcription factor is translocated into the nucleus, that's when gene expression follows and hypertrophic response follows. And so what we can show with our model is that as a function of the ETC, the calcium signal from endothelin-1, this is the sort of excitation transcription calcium signal. So excitation transcription coupling calcium signal. As a function of concentration, we can see that there is translocation of NFAT into the nucleus. So when there's no signal, we have very little NFAT in the nucleus, and when there's a larger signal, we have a lot of NFAT in the nucleus, and gene expression will result. Now these signals are several orders of magnitude smaller than these signals. So these signals are the beat-to-beat calcium, which is occurring to couple electrical activation and mechanical contraction. So the question is, well, what happens if we superimpose these ECC, excitation contraction coupling signals, on top of the system when we've got these signals for the hypertrophic pathway? And the answer in multicolor is as follows. So if we increase the pacing frequency, we still get translocation of NFAT to the nucleus. If we increase pacing frequency, then the amount of the sort of residual amount of NFAT in the nucleus does increase, but we still can see this signal over and above the beat-to-beat calcium. So what this is telling us is that a system where we have both of these pathways going on at once in the same space can distinguish a signal from the hypertrophic pathway from a signal from the beat-to-beat excitation-contraction coupling. So really this is telling us that the cell, so it's telling us that calcium can play multiple roles, and we knew that was the case, but it's really telling us that this signaling pathway, this apparatus is acting as a form of a low-pass filter, so it can essentially filter out the higher frequency calcium oscillation from EC coupling and leave you just with a low-pass filter. So it filters out the higher frequency signal and just gives you the slower, smaller signal from hypertrophy. But really what I want to use this to illustrate is that mechanistic modeling allows quantitative and predictive models to be built, and I would argue this is better than the hand-waving that Mike Berridge was talking about. We can actually get some quantitative leverage on these kinds of questions. And I think, as I said before, it gives us the confidence that we can extrapolate, we can predict and we can extrapolate beyond just the data that we used to build the models. But, and there is a sizable but here, when we just published that work, some people in Cambridge published a study in which they proposed a quite different view of what's happening. And their view was that actually there is spatial separation here. So this is the same figure I showed before. Their view is that the IP3-induced calcium release, so the hypertrophic calcium release, is just located near the nucleus. So it's really hidden away in a part of the cell that just doesn't see this excitation-contraction coupling calcium signaling. So they're suggesting that there is calcium released near the nucleus, and that's one kind of calcium, and there's calcium released from the calcium stores which binds to the myofibrils and causes contraction, and that's another kind of calcium, and these are kept spatially separated. And this is the prevailing view, so this is the experimentalist view of what's happening here. And so what we've now set out to do in collaboration with this group in Cambridge at the Babram Institute, which incidentally is where Mike Berridge, I think he's just retired from there, but that's where he was based. So with the group there from Lou Roderick and others. So we're now looking at building models of the spatial structure of the cell, so we can really answer this question as to whether this spatial idea works, so whether calcium can be kept in different parts of the cell, given that it diffuses very easily, and what difference that may have for this pathway or the interaction of these two pathways that we've been trying to follow. So to do that, we need to build models which have the geometry of the cell, and so we use EM tomography data, and we build, we reconstruct not the whole cell, but certainly reasonable regions of the cell. So I'm just showing myofibrils reconstructed from EM tomography. And we need to know where the calcium handling proteins are. So we use super resolution confocal microscopy to locate the key calcium handling protein. So this image here from Christian Solar's lab in Auckland, with whom we're collaborating. So the red dots here are little clusters of the RYR protein. So this is the calcium release channels for excitation-contraction coupling. And so we're looking at the sort of bluey-green stain, caveolin 3. This is telling us where membrane is. So this is the cell membrane in a cross-section of a myocyte. The sort of invaginations you see here are the T-tubular structure, so this sort of network of membrane that goes into the myocyte, goes into the muscle cell. And the locations of the red dots, the locations of the RYR proteins, in fact these are clusters of RYR proteins, sitting in that structure. And so from that, again, computationally we can reconstruct this geometry. So we reconstruct the cell membrane, the T-tubular structure, the locations of the RYRs, their calcium release units, and other key calcium handling proteins in this geometry. And I'd hope to be able to show you a pretty movie of what happens when we then simulate calcium handling on this system, but I'm just going to have, we're going to have to make do with some still images which aren't anything like as pretty or as illuminating, but safe to say that we can then solve the reaction diffusion equations for calcium diffusion on this geometry with calcium being released at these particular sites in response to activation. So what's clear here is that we need to do more than just ordinary differential equation models. We need to start thinking about the structure of the cell, bringing that detail about the location of these proteins if we want to get to grips with some of these very basic science questions about, in this case, how the cardiac myocyte works. Okay, so that was looking at signaling mechanisms in cardiac cells to try to motivate the need for mechanistic modeling and to give you also some motivation, I hope, for why we need to think about the structure of these cells rather than just some sort of a mixed bag where everything interacts with everything else. The second example I want to talk about is in cancer, is prognostic markers in cancer. And this is really to allow me to introduce network modeling. So I'm going to talk briefly about gene regulatory networks. So here's a very sort of schematic view of cellular organization. We've got a level of genes, and these genes make proteins, encode proteins, and so we have a level of proteins. And these proteins may be transcription factors which regulate genes, or these proteins may be enzymes which regulate metabolic processes. So we've got a level of kind of metabolic activity. And some of these metabolites may directly or indirectly regulate transcription. So we have this kind of complicated sort of hierarchical picture of what's happening in a cell. Genes encoding proteins, proteins doing things, including regulating metabolic pathways, interacting with each other, and ultimately regulating gene expression again. Now, we can, well, conceptually, we can project all of this information down onto the level of the genes. So we can project, for example, if gene 1 makes a protein 1, and protein 1 regulates gene 2, then actually we could say that gene 1 was regulating gene 2. So the idea of a gene regulatory network is to say we can project all of these cellular pathways down onto the level of genes. And of course the reason we want to do this is because we can measure gene activity. We can measure gene expression. So gene regulatory network. We can measure gene expression using, well, several technologies now, but we've mostly been using gene microarray technology. So we can measure which genes are being expressed at a given time or under different conditions in the cell. So we can follow things over time, or we can, by using a sequence of microarray studies, or we can perturb the cell in different ways in the way that I suggested before and measure how the cell's responding to different perturbations. And so we've been using siRNA to knock down particular genes, which are thought to be important in cancer, and to see how the cell responds, so that we can then try to build a picture of what was the underlying regulatory network, so we can learn more about the regulation of cancer cells. And actually one of the things we've been doing is a very simple idea, which is just to build these networks from our data and identify highly connected genes, which we might call hubs. So genes which seem to be involved with lots of other genes, regulating lots of other genes, and to see if they say anything interesting about cancer. So I'll briefly talk about an example of this applied in melanoma. So I won't talk much about melanoma other than to say melanoma is skin cancer. What we've been doing is taking a cell line, okay? So not ideal, but we've been taking a cell line, hitting that cell line with siRNAs, which are knocking down genes that we think are key genes in melanoma biology, collecting microarray data for each one of those. And so this is our perturbation step. So using multiple different siRNAs, knocking down key genes, recording using microarrays how the cells respond. So that's perturbing the system in a number of different ways. And then using technologies which I'm not going to go into in any detail, but technologies to take those data and to reconstruct networks which suggest interactions between different genes. So this is purposefully slightly blurry and a bit small, so you can't see the names of the genes because I'm not allowed to tell you names of genes that we found. But safe to say that there are some genes that appear to be very interesting. So for example, this is a hub gene meaning that it's simply very highly connected, that it's connected to lots and lots of other genes as identified in this network. And if we look at patients now, so if we have microarray data for patients, melanoma patients, and we look at these Kaplan-Meier plots which tell us about survival, so proportion of patients alive after a certain number of days, if we look at dividing that population of melanoma patients into ones whose expression of this particular gene was very low, we see that they survived. They survived for a long time compared to the rest of the population. And if, on the other hand, we look at those patients within the population whose expression level of the gene was very high, then we found that they didn't survive very long. So this is a kind of an approach to systems biology, which is, I think, increasingly common, which is to take some data, to perturb a system, to take that data and to build a network which tells you about relationships in the data. There's no mechanism here at all, okay? But it tells you about relationships in the data which help you to identify molecules which may be useful for prognostics. So there's no mechanism here, there's no way we can predict very much here. about the biology, but certainly it seems to be telling us something useful about the clinical outcomes in this case. So we've been interested in this kind of approach, and we've been interested in making this kind of technology available to biologists. And so we've recently worked to produce a software system that we hope is going to be usable by biologists. And so what this software does is take in, for example, microarray data, or it doesn't have to be microarray data. It processes that data in various ways, so normalization, standard kinds of things you'd do, sort of bioinformatic processing of microarray data. It then processes these data for network inference, and we are not in the business of only using our own tools. There's a lot in this area of research where people will come up with a new algorithm and say our algorithm is so much better than everybody else's. What is actually really important here is that we use all the tools that are available, and so we've made available a lot of the different technologies for inferring these networks, including our own, but not technologies from lots of other groups. And then importantly, so we have this workflow which allows this to be done in a very straightforward way. And then importantly includes lots of visualization tools and analysis tools for then looking at the results of these kinds of network inference, including the networks themselves, but including lots of other ways which I won't go into as to how you can visualize these networks and try to learn something useful from them. And so we've used technology called gene pattern, which comes from the Broad Institute at MIT, to build this kind of workflow into a format which is readily usable through online resources. So this has recently been published. So we've used some of this technology in the third example that I want to talk about, which is looking at signaling pathways in a tissue, okay, in situ. And the reason I want to talk about this is because I think it reveals quite a lot about the dangers of looking at data drawn from databases, drawn from multiple different cell types. So first of all, I'll say something very brief about the skin, about the epidermis, where we've been focused. So the epidermis is a barrier. It's a barrier to environmental insult, so stopping stuff getting in, but also a barrier to stuff getting out, so minimizing water loss. And the way that the skin achieves this is through its structure. So if you look at the epidermis, it has a particular structure in these cells which start from a basal layer. They move outwards over time, and they form this sort of cornified layer here, which is essentially dead cells which form that barrier. Now this arrangement of cells, this arrangement of the tissue, is a differentiation gradient. So the cells start here, they divide and they divide and they get moved out, and as they move in time or in space, however you like to think about it, they change, they express different proteins, and ultimately they form this cornified barrier there. And this process, this differentiation gradient, is thought to be controlled by a MAPK signaling pathway, so a cascade of signals, so the MAPK-ERK signaling pathway. And so we're interested in saying, well, okay, this kind of signaling pathway is commonly studied in cell lines, in isolated cell lines, cell cultures. But we want to know, well, what does this pathway actually look like when it's regulating this process in time and space in the skin? Now, if you go and look at the literature and you find out what is known from the literature in terms of interactions between these key molecules, and you restrict your search to, for example, human epithelium or mammalian keratinocytes, then you pull out a huge number of potential interactions. You pull out a huge number of interactions. So this is dividing those interactions into proteins sat in the plasma membrane, proteins in the cytoplasm, proteins in the nucleus. And each of these colored diamonds is a protein or phosphorylated version of that protein. And you can't see the names, but it's essentially these and some other ones. And each of the arrows says that there is in the literature or in a database that we've used, there are identified interactions between these proteins. And so we want to ask, well, is this actually a good model at all for what's happening in the skin, in the tissue? And so the way to do that is that we need to then measure the abundance of these proteins in the skin itself. And once we've measured the abundance of these proteins, we then need to apply our network techniques to try to develop a network with which to compare to this one. And so that's what we've done. So I will very quickly tell you about decisions we made about measuring protein abundance, because a lot of this I think will be familiar, but there are lots of techniques by which you can measure protein abundance. Western blotting, for example, is pretty much necessarily a tissue averaged measure. And so that's not of any use to us because we want to know protein abundance at a specific location in the tissue. We don't want to know protein abundance across the whole of the epidermis. We want to know at a particular point in space, what is the protein abundance. So that's no good to us. And mass spec is promising, but also no good to us just because we can't get the spatial resolution that we need. And so really this only leaves us with confocal microscopy. So very now conventional, traditional approaches of immunofluorescence and confocal microscopy. But that gives us spatial information and it gives us resolution which is actually subcellular. So we can resolve between different cellular compartments. We can resolve expression of proteins in the membrane, proteins in the cytoplasm, proteins in the nucleus. And so I have a slide just showing you that if this is not kind of old hat to you. But safe to say that some of the key proteins we're looking at here, this is looking across the skin. This is human skin, by the way. Looking in sections across the skin and looking at expression in the nucleus or expression here in the cytoplasm, but not in the nucleus. Here's expression in the membrane, for example. And so from these kinds of data, we can then use this as a quantitative measure of protein abundance. And so we can use these kinds of images to build a picture where we now scan across the skin and we normalize across the skin. And we say, as a function of position, what was the abundance, what was the expression of a protein, in this case phosphorylated MEC. And the different colors here are several different skin samples from different patients that we had access to. And so we can look at a profile across the skin from the basal layer to the to the cornified layer, of the changes in protein expression as we move through the skin. And so this is looking in the cytoplasm and this is looking in the nucleus. And there would be one for the membrane if this protein was expressed in the membrane, but it's not. You can see it's not. You can see the sort of darker outline where the membrane is. So this, what's beautiful about the skin, of course, is that this is a spatial coordinate, but it's also a temporal coordinate because we're moving, the cells start in this basal layer and they move through the skin as they age and as they differentiate. So we've done this. When I say we've done this, again, this is this horribly unfair thing. I didn't do this. A very dedicated graduate student did this for 18 phosphoproteins with several different skin samples from several different patients. It was a bit of a tour de force in terms of collection of the data, and has measured and characterized the expression of these different phosphoproteins in these different cellular compartments. And so what we end up with is a lot of these kinds of data. These have all been nicely smoothed and normalized, but a lot of these data showing how the expression of these proteins changes as you move from, in our normalized spatial sort of measure of depth through the skin, through the epidermis. It tells you how the protein concentrations change. And so in order to then apply these network inference techniques, we then discretize, and we've chosen to discretize in a way which is consistent with the number of cells here. And then we apply the techniques which we'd previously applied, for example, to those cancer gene expression data sets. In this case, looking at this process as if it was a temporal process, which in a sense it is. So looking at how the information here tells us about the information in the next, can be used to tell us about interactions in the next spatial position. So by using this sort of dynamic argument, we can build networks. So this is the network I showed you before. This is the interactions reported in the literature collected from cells, from cell culture, and this is what we pull out. So we actually get a lot of very sensible looking, so I'll show you. I mean, the take-home message basically is that these two things don't really look anything like each other. There are many, many fewer interactions here. So I won't go into details, but this, the interactions here make biological sense in terms of the pathway, the sort of canonical pathway that we know about. But what I think this is telling us, and we need to go back and validate some of these interactions. Some of them are not what one would expect, so we need to go back and validate them. But I think what this tells us is that, and maybe this isn't a surprise, but only a fraction of literature or postulated interactions is actually relevant for this particular example. So it's come from the literature analysis came from relevant cells. It came from human epithelial cells, but only a fraction of those interactions seem to be relevant for what we're seeing in our samples. And so not only that, but also if we take, so what I told you is that we are very careful to make sure that when we build our network, we are looking at cutting, if you like, sort of computationally cutting our tissue up into these strips, and we're looking at how this network, sort of, how these interactions move through the tissue. And if you don't do that, if you scramble all the data up together and you treat it as if it was isolated cells, then you get something quite different. And so it's also telling us that actually the spatial structure of the tissue here is really important for understanding what the underlying network controlling, in this case, cell fate is. And so I think we learned that network data that's collected out of context of a specific tissue, or which has removed the spatial structure of that tissue, again, may be very misleading when it comes to looking at the underlying biology that it tells us about, or it may simply not be very useful for telling us about the biology in real tissues. So I've told you about mechanistic modeling and why I think mechanistic modeling is important, rather than simply representing, for example, networks. But I've also told you about networks and how we go about inferring them. And I've tried to argue that tissue context is really important in pursuing this kind of research. And I hope I've also covered the uses of systems biology in basic research, so just approaching basic biological questions. Showed also that there are major applications in translational research, of course, but also we can just use this for discovery science. So we can use systems biology approaches just to learn more about some of these systems. So I'm going to finish there, but with acknowledgements to the people that I've worked with. So this is people from my group in Auckland. So the cardiac work was done mainly by, initially by Mike Cooling, who finished his PhD with me, and is now being picked up by Vijay and Greg Bass in my group. The network inference work, the cancer work has been done by Yikan Wang. The development of the software was done by Dr. Daniel Hurley. The cardiac work was in collaboration with a lot of people, including Christian Solar in Auckland, Lou Roderick in Cambridge. The network inference work is being done in collaboration with Chris Print, who's a colleague in the medical school in Auckland. And the skin work was done by Joe Curzons, who's he's one of these amazing graduate students who comes along and is equally adept at doing the experimental work and doing the modeling work, and I need to clone him pretty urgently. And that's done in collaboration with Rod Dunbar in the School of Biological Sciences in Auckland and Mark Jacobs in the Bioengineering Institute. And of course, thanks to the people who fund our work. So thank you all.